In the introduction, I talked about two RNA molecules. One that it was the messenger RNA, and another one that is called the tRNA. And there are many more molecules of RNA involved in the transcription and translation. But for this class, I just want to tell you that there is another one that is important, that is the ribosomal RNA, and we're going to talk about that. I'm going to explain the main function of the ribosomes using this diagram of your textbook. But first, I want that you notice that there are two RNA molecules. The transfer RNA that comes from here, that as you can see is attaching to amino acids. Here an amino acid is attaching to a transfer RNA and is going to go inside of the ribosome that you can see it in brown. And the other molecule that is very important is the messenger RNA that also goes inside here and lives here. And the transfer RNA will live here. So they will both go inside, they will couple the codon and anticodon, and then will both live. So what is the main function of the ribosomes? The ribosomes facilitate a specific coupling between the tRNA anticodons with the messenger RNA codons in the protein synthesis. And maybe here is not very clear. I'm going to show you another figure, but the ribosome is composed of a large subunit and a small subunit. Here, the one below is the small subunit, and the one on top is the large subunit. And again, this is a diagram, but I'm going to show you, as usually, a computer simulation of what scientists think that the ribosome will look like. So here you can see the computer model of a functioning ribosome. And as I said, there are two subunits, a large subunit in blue here, and a small subunit here in white. Here you have, as I said before, the tRNA molecules and the messenger RNA that both are going to get inside. And here you can see the growing polypeptide, and here you can see the exit tunnel where the polypeptide is going to go outside. In this slide, I'm going to talk about the three binding sites for the tRNA, and I'm going to use this diagram from your textbook to explain that. And I just remind you that you already watched a video talking about this, but so I'm going to go and talk very generally about key points. So for your exam, remember that this one, it will be the messenger RNA binding site. The messenger RNA is going to come here. Here, if you remember, this is the large subunit, and this will be the small subunit of the ribosome. And you have here 1, 2, and 3 binding sites for the tRNA. The first one will be called A, the second one will be called P, and the first one will be called E. And in the A, you have the tRNA with the amino acid here. So they will go and attach to this binding sites. Later, in the P site, if you remember, this is the site where you have both the codon and the codon attaching. And as you watch in the video, the amino acid will be transferred here and the polypeptide will start growing. So this is the P comes from that polypeptide. And finally, the E is the exit one, the exit binding site when the tRNA leaves the ribosome. So let's recap all the things that we just said. So first thing that I told you is that the messenger RNA has a binding site. Then I told you that there is a large and a small subunit in the ribosome that there are 3 binding sites. So let's first, second, then I told you there were 3 binding sites. The first one will be A, second one will be P, and third one will be E. So the messenger RNA and the tRNA will go in this direction. The P site is hold the tRNA that carrying the growing polypeptide change. So this is the P site. The A site hold the tRNA that carries the next amino acid to be added to the change, and this is the A change. And finally, the E site is the one where the tRNA will exit. And remember, the P sites has this name because of this, because it's the peptidyl RNA binding site, while the A site is the amino acyl tRNA binding site. And as I just said, the exit site is the E site and is the place where the discharged tRNAs leave the ribosome. So very quickly, there are 3 binding sites. A site, here, if you see, you have the tRNA, and here you have the next amino acid that is going to be added to the polypeptide chain. So it's ready to be added, but it's only added when it moves to the P site, that you have this growing polypeptide here, and the next one that is going to be added, it will be this one that it will add in a second. And when the amino acid is added in the polypeptide chain, the amino acid, it will go to the E site, as you can see here. And as they don't have any more an amino acid, because it was put it here, now it is released. Now let's try to answer the following questions. Maybe you can stop the video and you can try to answer on what is the function of the RNA. Try to look what is the function of the tRNA based on what we said. This is the function of what? Plays a catalytic ribosome roles and structural roles in ribosomes. So try to, if you don't find the question, you can go to your textbook. What is the primary transcript? And we haven't talked about this, but you can find it in your textbook. So please stop the video and try to find those answers. Now, these are the answers. And you can go and read it and make sure that you're right. In particular, we didn't talk about this in the video lecture, but you can look for that in your textbook.